Hello everyone. This is Kendra Vidyalaya Sangathan JIT Bhubaneswar's initiative. We are presenting digital videos for chapters in class 11th and 12th.
Students, we will continue to the third part of our existing lecture in biotechnology, principles and processes. Today's topic for discussion is tools in biotechnology, cloning vectors and making host competent. As you are aware that the chapter is divided into 5 learning units, we have already completed the 2 and we are in the third unit. In the ladder of concepts, we are at the third rung, tools of biotechnology, cloning vectors, making host competent. What is expected out of you before we take on this lesson? You should know the basic structure of DNA as double helical structure. How did we create recombinant DNA? You should also know the difference in nature of cell wall of different organisms and basic of DNA replication mechanism. You should also know a bit about chemical nature of DNA that you have read in your earlier chapters. After completing this lecture, you will be able to explain the structure of plasmid, describe the types of cloning vector and its characteristics, explain structure of typical cloning vector PBR322, elaborate the various mechanisms that makes a host cell competent to accept recombinant DNA. In extension of our understanding of tools of biotechnology, we have already seen there are enzymes, including cleaving enzymes, lysing enzymes, synthesizing enzymes, and joining enzymes. Then we had gel electrophoresis, the technique that separated the cleaved DNA. In this lecture, we will study about cloning vectors or vehicular DNA, which will include plasmid, cosmid, and bacteriophage. We will discuss plasmid at stretch, and then ways of making host competent to receive the recombinant DNA. It has got chemical method, electroporation, gene gun, and microinjection. First of all, we'll talk about cloning vectors. Cloning vectors includes plasmids, cosmid, bacteriophage, phasmid, YSC, yeast artificial chromosome, and VSC, bacterial artificial chromosome. And ways to discuss ways to make the host competent, including the chemical method, electroporation, the gene gun method, and microinjection method. Let us first of all talk about cloning vectors. What are these cloning vectors? These are the vehicular DNA that can be also called as extra chromosomal replicons of DNA, which can be isolated and can replicate independently of chromosome. That means they are mainly present in bacteria and in some yeast. They do not depend on the chromosome for replication. Please add another concept. If a vector replicates in 2 hosts, it is called as shuttle vector. So we have got certain types, plasmids. They are autonomously replicating extra chromosomal circular DNA, generally found in bacteria and sometimes in yeast. And this term was coined by Joshua Lederberg in 1952. The cosmids, they are the hybrid plasmid that contains lambda phage cos sequence. Lambda phage is a virus. Cosmid, you can break it up. It will have cos sites and plasmid sites. So we call it cosmids. And DNA sequence are originally from lambda phage. Bacteriophages. These are the viruses that infect bacteria and are called as bacteriophages. They are used to transfect bacterial cells. We have seen in the previous lecture, bacteriophage, which delivers the DNA inside bacteria, and bacteria has got restriction system that prevents it. Hence, the restriction enzyme was called as restriction endonuclease. There are some other vectors Plasmid, YSE and BSE. Basically, plasmids contains small number of bases, whereas if large DNA has to be inserted, we have to rely on bacteriophage, YSE and BSE. These were used in human genome projects. So let us focus on plasmids. It was discovered by Joshua Lederberg in 1952. They are autonomously replicating, and it can occur naturally or can be made artificially. The one diagram that we see here is of PBR322. What is this PBR322? PBR, P for plasmid, BR for the name of discoverer, Bolivar and Rodriguez, and 322 is this number that they have discovered or synthesized, because it's just like giving roll number to the students, right? So it is around 2700 base pair long. There are commonly used vector systems like bacterial plasmid, bacteriophages, cosmid, yeast artificial chromosomes, TI plasmid, that is tumor-inducing plasmid, eukaryotic viruses such as baculoviruses and S40 viruses and even retroviruses. Now let us talk about characteristics of a cloning vector. You can find there are certain numbers, certain names given. We have already dealt with nomenclature of restriction enzymes, so we can easily find out these names are nothing but site where these restriction enzyme can cleave. Let us find out what are the characteristics that we need to have. An ideal vector should have, number one, ORI, origin of replication site. This is one site which controls the copy number and also ensures that whatever sequence is inserted, it also gets replicated. It should have protein, replication protein synthesizing gene, which helps the DNA polymerase to recognize and start the replication process. Selectable markers, as you can find here out, just inside the PST1 sequence and BAM-H1 sequence, there are two sequences, ampicillin resistance and tetracycline resistance. These sequences provide resistances to bacteria against the antibiotics, and they are used as selectable markers. Why selectable markers? We will discuss them because these are the one that ensures selection of recombinant from non-recombinant. Restriction sites, as you can find out, different sites at which the foreign gene will be inserted, and the size should be preferably smaller to help easy handling and replication. Now, when we talk about origin of replication, this process marks autonomous replication in the vector. ORI is a specific sequence of nucleotide in DNA where replication starts. When a foreign DNA is linked to this sequence, then along with the vector replication, the foreign DNA or the desirable DNA also gets replicated. This part also controls the copy number, that how many copies will be created. So very, very important characteristics for a cloning vector is ORI. Then the selectable marker. These are the genes whose expression allows one to identify the cells that have been transformed or transfected with a vector containing the marker gene. A marker gene is used to determine if a piece of DNA has been successfully inserted into a host organism or not. Normally, the gene usually encoding for resistance to antibiotic is used as selectable marker. What happens? Whenever any gene is inserted within this sequence, the resistance is lost. Hence, once you plate these colonies on that particular antibiotic, they may not be able to survive. This process may be a bit cumbersome. Hence, there is another concept that we'll discuss shortly, which is called as alternate selectable marker. Now we talk briefly about restriction sites. These are the sites that allows cleavage of specific sequence by specific restriction in the nucleases. Restriction sites in E. coli cloning vector PBR322 includes HIND3, EcoR1, BAMH1, SAL1, PVU1, PST1, CLA1, etc. More cloning sites will generate more fragment. So cloning vector should have one site for cloning. It should not be contradicted because this is artificially made cloning vector, hence it facilitates working for different types of restriction enzyme. Now, as we can see out, these sites, they are named as per the convention into derived from Haemophilus influenza, D strain, and third in the order of discovery. It will remind you of the nomenclature convention as we have used in case of restriction enzymes. So the main characteristics of plasmid containing vector in PBR322 are they'll have some restriction sites like HIND3, BAMH1, SAL1, PVU2, PST1, PVU1, EcoR1, and CLA1. Selectable markers, they are antibiotic resistance genes for ampicillin resistance and tetracycline resistance. ORI and ROP, that codes for protein which are involved in the process of replication of plasmid. This diagram is very, very important. Must practice twice or thrice because in examination it is generally asked. And I have already described why it is called as PBR322. Plasmid of Bolivar and Rodriguez, and the number is 322. PUC19 is another commonly used cloning vectors that is generally used. It is developed in University of California, hence called as PUC19. TI plasmid, tumor-inducing plasmid. It is isolated from a bacterium, Agrobacterium tumefaciens, which is used to transfect plant cells. And this is also called as natural genetic engineer. Why? Because it can push the TI DNA to plant genome and stimulate cell division, thereby causing tumor. So there are ways whereby we have disarmed this pathogen and inserted a desirable sequence so it can be delivered naturally to the plant. RA plasmid, root-inducing plasmid, as isolated from a rhizogen, bacteriophages that naturally infects bacteria. These are the most efficient than plasmids for cloning large DNA insert. Cosmids and plasmids are another option, and a part of that bacterial artificial chromosome and yeast artificial chromosome. They are large side artificial chromosome used to clone genes in human genome project. There is a representation of TI plasmid. So now we talk about the selectable markers. So we have seen that there are two selectable markers in PBR322, ampicillin resistance and tetracycline resistance. So selectable markers helps to select the host cell which contains the vector transformant and eliminating non-transformant. So on inserting a sequence in antibiotic resistance sites, the resistance will be lost and bacteria will die in the medium containing that given antibiotic. Take for example, if a sequence is inserted in tetracycline resistance area, then it will lose the resistance for tetracycline, and when they are placed in a medium containing tetracycline, what will happen? Bacteria will not be able to survive. But selection of recombinant due to inactivation of antibiotic is a cumbersome procedure because it requires simultaneous plating on two plates having different antibiotics. That may not give a very effective results. Hence, alternate selectable markers are devised, and these are used to select transformant. Right? As I have already mentioned, there is a procedure called as insertion inactivation. So, if insertion of recombinant DNA is done in the coding sequence of enzyme beta-galactosidase, that leads to inactivation of the enzyme, and that is called as insertion inactivation. The recombinant do not produce a blue-colored colonies in presence of a chromogenic substrate, while the non-recombinant continues producing blue color. Why? In non-recombinant, there is no insert in the sequence of beta-galactosidase. Thus, the production of substrate that converts into blue goes on naturally. But in case where the insert has been placed in the sequence of beta-galactosidase, this does not take place, right? Thus, coding sequence of beta-galactosidase is a better selectable marker. Now let us see. We have got the recombinant DNA, and what are the ways to make them competent? That means they should be able to accept the plasmid. There are some methods like chemical method, physical method, and vector method. Already we have discussed about vectors, how agrobacterium tumefaciens transfers or bacteriophage transfers. First of all, the host like bacterial cell is made competent to be transformed with the recombinant DNA. This is a vectorless method. Cell wall is removed using appropriate enzyme like in E. coli, fungi, and plants. What happens? We treat it with certain enzymes like lysozyme, like chitinase, like cellulase. Then cell is treated with divalent cations like calcium and magnesium to neutralize the negative charge of DNA and negative phospholipid charges in the membrane. Then a heat shock is given by placing the cell at 0 degrees Celsius and then taking it back to 42 degrees Celsius and again bringing them back to 0 degrees Celsius. This increases the cell membrane permeability. So this is what happens, and now the cell is in a position to take the recombinant DNA. There are other ways for vector-based gene transfers. Chemical method, protoplast, means the cell without cell wall, that is incubated with the gene and kept in PEG, polyethylene glycol, that allows the entry of gene into the host. Another method is called as electroporation. Cell is kept in medium which is supplied with high voltage for a short time. And this results in creation of tiny pores through which the gene enters, and afterward the pore closes. Gene gun method are also very common, generally for the plant cells. So DNA coated on the gold or tungsten particle, the size around 1 to 2 micron, and they are shot at the rate of 1400 feet per second to make particle reach the host nucleus. In case of animal cell, there is another technique used called as microinjection. Using microscopy, a micropipette is used, and microinjection transfers the gene in animal cells. So very important for us to understand that the host has to be competent in order to accept the recombinant DNA. So we have read in this chapter, the biotechnology in recombinant DNA includes manipulation of DNA and then inserting into host for its expression. Cloning vectors are extra chromosomal replicons of DNA which can be isolated and can replicate independently of the chromosome. Cloning vector should have ORI, ROP, selectable marker, restriction sites, and small size. The commonly used cloning vectors are plasmids, cosmids, phasmids, bacteriophage, BSE, and YSE. There are selectable markers and alternate selectable markers that is mainly used to select transformant from non-transformant. And at last, the cells are being made competent to allow them to be transformed with foreign DNA. And we have seen how they are made competent by treating them with solution of calcium chloride, like giving them divalent cation and then giving them a heat shock, whereby they are placed on 0 degree, moved to 42 degrees, and then brought back to 0 degrees Celsius to allow cell membrane to permit the gene to enter. There are other methods of gene transfer without a vector, such as chemical method, electroporation, gene gun, and microinjection method. Now let us talk about some questions. To check your understanding, the given figure of diagram represents vector PBR322. Which of the following option gives correct identifier? ORI represents the original restriction enzyme. ROP, reducer osmotic pressure. Hint 3, eco-R1s are selectable markers. Ampicillin R and tetracycline R are antibiotic resistance genes. A gene whose expression helps to identify transformed cell is known as vector, plasmid, structure gene, selectable marker. Which of the following is not a feature of plasmid? Transferable, single-stranded, independent replication, circular structure. Next question. The colonies of recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria because of insertional inactivation of beta-galactoside in recombinant bacteria, inactivation of glycosidase enzyme in recombinant bacteria, non-recombinant bacteria containing beta-galactosidase, insertional inactivation of alpha-galactosidase or beta-galactosidase in non-recombinant bacteria. Bialistic or gene gun is suitable for dissolving pathogen vector, transforming of plant cells, constructing recombinant DNA by joining with vectors, and DNA fingerprinting. If you have opted for your answers, let us see what are the correct responses. Yes, for the first, ampicillin resistance, tetracycline resistance, antibiotic resistance genes. Next, selectable markers, single-stranded, insertion activation of beta-galactosidase recombinant bacteria, and transformation of plant cells. If you have answered them correctly, you have understood the concept well. Try taking some home assignment. Take this question, write it neatly in your notebook and show it to your mentor. There are some further readings. You can visit these sites, have a look at the animations. So up till now we have seen the principles and overview of biotechnology. Then in tools we have read about restriction enzymes and gel electrophoresis. And in this part we have read about cloning vectors and how to make the host competent. So in the coming part we'll be talking about the processes of recombinant DNA technology, and we'll discuss about isolation of DNA, cutting of DNA and gene amplification using polymerase chain reaction. So keep tuned. This video lecture was presented by Mr. Rajiv Ranjan Singh, PGT Biology, Kendra Vidyalaya, Air Force Station, Kumbhagram of Silchar region. This is Jit Bhubaneswar presentation. Thank you for viewing. Keep watching.